Hello, everyone, and welcome to the latest Genetic Engineering and Biotechnology News webinar. Our presentation today is entitled Therapeutic Antibody Discovery by Mammalian Display. I'm Jeff Bogaliskas, Technical Editor for Gen, and I'll be the host for today's webinar presentation. Accelerating the drug discovery process, especially for biologics, not only helps companies get potentially life-saving therapies to market faster while increasing their revenue, it can also be a major boon for researchers, acting as the impetus for innovation. For instance, the use of gene editing technology has allowed for the direct integration of antibody genes and creation of mammalian cell libraries, leading to single antibody per cell constructs. Consequently, this has enabled the construction of libraries containing millions of stable cell lines displaying monoclonal IgG formatted antibodies on their surface, from which novel high-affinity binders can be isolated and in turn fast-tracking the drug discovery pipeline. Let's meet our speakers for this webinar who will describe how an innovative antibody discovery technology can save time and effort by addressing issues during the initial drug discovery phase. Additionally, we'll hear how MaxCyte's innovative electroporation technology can be utilized during therapeutic antibody development to streamline the drug discovery process. John McCafferty is CEO and founder of Iontys. Dr. McCafferty was the pioneer and inventor of antibody phage display, which has revolutionized monoclonal antibody drug discovery and is widely used in the biopharmaceutical industry for the development of therapeutic antibodies for a variety of diseases. In his presentation today, Dr. McCafferty will describe the science behind a novel technology developed at Iontys and how it's utilized in the antibody discovery process. Payal Rochaudhary is a field application scientist at MaxCyte. Dr. Rochaudhary will tell us about the advantages of MaxCyte's delivery platform and how it can be used from enhancing basic research projects to large-scale bioproduction applications. So before our speakers get started, I want to encourage the audience to submit questions for our Q&A session after the last presentation. We'll try to get to as many questions as we can, so simply type your question into the box below the presentation screen and hit submit. All right, let's get our webinar underway. John, the audience is all yours.
Good morning. My name is John McCafferty and I'm the founder of Iontys, a biotechnology company based in Cambridge, England, devoted to the generation of therapeutic human antibodies. I'm delighted today to have the opportunity to tell you about the work we've been doing, particularly around the development of mammalian display, and I'd like to thank Genetic Engineering News and MaxCyte for that opportunity. So back in 1990, I was fortunate enough to be one of the founders of Cambridge Antibody Technology, now MedImmune Cambridge. And by the end of that first year, we had this paper in Nature describing for the first time the display of antibodies on filamentous bacteriophage of E. coli. What we were able to demonstrate was that we could clone antibody genes in such a way that when a filamentous phage particle was produced from that cell, that it displayed the encoded antibody on the outside with the antibody gene itself encapsulated within the bacteriophage particle. And in that way, we had created a link between the gene and the binding molecule which it encoded, and we were able to demonstrate that we could actually isolate specific antibodies from large mixes. So over the years we've been very excited and still are about the power of phage display to start with a large naive library of antibody genes, typically in the 10 to the 10 territory, and go from there through rounds of selection to finish up with a drug candidate. And in fact, one of the first molecules to come out from CAT was Humira, or at least a molecule that went on to become Humira. We found with this in vitro system that we indeed had power to push affinity, push stringency to generate the highest affinity antibodies from a population. Also in some way, because it's an in vitro system, we were able to direct specificity and ultimately generate a large panel of human antibody molecules that we could then screen for specific function. And being this in vitro system, We could create large libraries, even derivative libraries, from an initial starting molecule and go on and generate better and better molecules. So with such a great system in place, why bother to set up an alternative such as a mammalian display system, which we're talking about today? Obviously it's nice, since antibodies are ultimately going to be produced in an IgG format in mammalian cells. It's nice to be able to work the discovery process using that same format and those same cell types. In addition, with phage display, we start off with libraries of 10 billion or so clones, but ultimately end up screening perhaps a few hundred or a few thousand clones by ELISA in the first instance. And when we get a strong signal in those ELISAs, we can't tell or be sure whether it's down to high expression or high affinity, or in some cases dimeric forms of the antibody. And so to be able to work directly with an IgG format and screen by flow cytometry, we have the potential to screen millions of output clones in a relatively short period of time. We can perhaps anticipate what's going to happen in terms of expression of the soluble antibody as well, or certainly if an antibody is going to eventually be a very poor expressor, we might hope to anticipate that by looking at the expression level on the surface. But going beyond that, we believe that we can anticipate potential additional developability problems that emerge once one starts concentrating antibody up to a high level, and we'll come to that. So the challenge, however, in making mammalian display libraries is to find a way that allows you to make large libraries on the one hand, but large libraries where there's just one antibody gene in every cell. And the solution we've come up with is to use homologous recombination. About 20 or 25 years ago, people showed that if you carry out homologous recombination but initially cleave the genome at the site that you're driving this homologous recombination, that you massively increase the efficiency of that process by about 40,000 fold. And in that initial study, people had pre-inserted a meganuclease site into the genome. Meganucleases recognize 18 base pair sequences, and so it's either very rare or absent in the genome. And in this case, by introducing a meganuclease plasmid at the same time as a donor DNA is given, we're able to push up the rate of homologous recombination. And in the years that followed, people have generated site-specific nucleases in different ways. For a period, people are working with zinc finger nucleases. This was followed by the development of tail nucleases, where one is able to actually build a DNA recognition element from monomeric units by assembling them together, and one works with a pair of these tail nucleases to effect double-stranded cleavage at the site in the genome that you're interested in. This was first described maybe 5 or 6 years ago. Several years ago, of course, CRISPR-Cas9 technology was described, wherein one no longer has to build a specific protein to recognize a DNA sequence, but rather one can do it by generating a small guide RNA. So we have worked with all of those nuclease-directed systems, but most of the work that we've done in the early days was using tail nucleases, and more recently we've demonstrated the capability of CRISPR-Cas9 as well. We have a couple of tail nucleases that target a particular site in the human genome called the AAVS locus. It's basically a large intron of around 4.5 kilobases. So now we transfect the donor plasmid shown on the second line along with the DNA encoding a couple of these tail nucleases. The donor plasmid has relatively short homology arms of around 700 or 800 base pairs to drive the targeting of the incoming plasmid. We are typically using the MaxCyte electroporation system, which is a flow system wherein one can transfect up to 10 to the 10 cells in the course of about half an hour. And it's designed really to leave the cells viable at the end of the process. So it's ideal for the construction of the libraries that we work with. So we basically use the system to introduce the tail nuclease plasmids along with the donor plasmid. Following cleavage of the genome, we now get insertion of the cassette and finish up with the construct in the genome shown here on the bottom line. So if we look at this, we can see there's a promoterless blastocidin gene. It's promoterless, but it also has a splice acceptor sequence which allows splicing with the upstream exon that was already present within the locus. So this allows a clean up of the background, because in principle the blastocidin resistance will only occur if you get integration with an upstream exon in frame with that. The other part of the insert is this dual promoter cassette expressing an antibody light chain and an antibody heavy chain. In this model experiment I'm about to show, we've taken an antibody heavy light chain gene encoding an anti-lysozyme antibody. So now we can look at a couple of things following this transfection and selection for blastocysts. So in this experiment we've used the donor plasmid shown in the previous slide encoding an anti-lysozyme IgG molecule and co-transfected along with a couple of tail nuclease plasmids directed towards the AAVS locus, transfected into suspension hex cells which have been drug selected for a couple of weeks, and we're now doing flow cytometry staining with anti-FC antibody shown on the x-axis and labelled lysozyme shown on the y-axis, and you can see that the majority of cells are indeed expressing a functional antibody, functional IgG on their surface, with something like 83% of cells being double positive. For the system to work well, ideally we want to introduce one antibody gene only into each cell, and that's what this experiment is illustrating here. So here we've taken a 50-50 mixture of two different plasmids encoding antibodies that either recognise FGFR1 or FGFR2, and these antibodies are truly specific between the two different forms and were described in this paper shown here. These were introduced into the system, drug selection carried out, and then the population stained with labelled FGFR1 or FGFR2. Again, we're using the MaxCyte electroporation system to introduce the DNA. Very efficient system for getting DNA in for transient expression. Up to 90% of cells will express. However, in this case, we're looking for an additional step, which is the correct integration of the antibody gene into a particular locus. To quantitate that, we take the cells and we select them on plates with blastocidin, and we can then calculate the number of cells that have become correctly and stably integrated. And in this case, we find somewhere between 1 and 5% of the cells go on within about a week or so to give us a stable integration. To determine whether we've got single copy or not, we're now using the labelled anti-FGFR1 and anti-FGFR2 to look at this. And what we can see from the dot plot shown here is that the majority of cells within the population are positive either for FGFR1 or for FGFR2. And as I've said, we can calculate that somewhere between 1 and 5% of the cells end up integrating an antibody gene. In this case, we're working with a model library of around 1 million clones as a result of transforming 40 million cells. But using the flow electroporation system from MaxCyte, we have the potential, therefore, to work with libraries at between 1 and 5 times 10 to the 8, using 10 to the 10 cells as the MaxCyte system allows us to do. So moving on from model experiments as shown in the previous slide, this experiment illustrates the use of the system to screen an actual population. In this case, it's an enriched population from 1 or 2 rounds of phage display. So we transfect the population within the donor plasmid, again go through drug selection introduced after day 2. And here we're showing that after about 8 days since the transfection, we can do max sorting and generate an enriched population, and we find that this population is now stable and can be grown continuously and remain positive or IgG expression. In this example, we've taken that population at day 13 and done FAC sorting based on antigen binding, in this case using 10 nanomolar PDL1 from a population selected on PDL1 by phage. And you can see here that this is indeed positive and stable. So following the selection on day 13, the population has grown for an additional 9 days before being stained. We can see in the dot plot on the bottom left that again using 10 nanomolar PDL1 as was used in the sorting, that the majority of cells are indeed positive. And in addition, we can see that by reducing the amount of antigen, and as shown in the other dot plots, that we can actually begin to drive the stringency of the selection. So really this is showing us that we have a stable population of antigen binding cells after only 8 days, and this ability to use max sorting to clean up the population is quite useful in terms of debulking the culture and shortening the need for drug selection. And also this experiment illustrates the ability to drive the stringency of the selection to generate higher affinity antibodies by pulling out with limiting antigen. So we have used the system described here to screen millions of output clones from a phage display population. We've also used it to do affinity maturation of a starting clone, either by chain shuffling or by oligonucleotide directed mutagenesis, and we've also combined that with IgG reformatting of clones that may have started as single chain FVs. And again, through the use of the system, we've been able to generate functional antibodies, for example, antibodies which block PDL1 function. There are, of course, several options for generating human antibodies, and some of these are shown here, each with their various pros and cons. But ultimately, whatever the source of the antibody, there's a need to actually turn it into a drug molecule for it to actually be developed into a product. And historically, this process of antibody discovery and antibody development have been dealt with within separate silos in pharmaceutical organizations. So really, development people have to understand or deal with whether it can actually be turned into a drug with cost-effective production, stable formulation at high concentration, and optimal pharmacokinetics and pharmacodynamics. I'd like to point out that whatever the source of the antibody, there are potential biophysical liabilities that may prevent this successful development. Just because an antibody has come from a so-called natural source, for example immunization, doesn't mean that it's going to be free from these liabilities. Nature hasn't evolved antibodies to be concentrated up to 50 or 100 milligrams per mil. I mean, certain sequence liabilities can also be designed out, such as glycosylation or amidation sites in the starting library. But the tendency for self-association or precipitation, leading to precipitation at high concentration, or a tendency for stickiness can't really be designed in this way. So we wondered whether mammalian display could possibly be used in a way to actually anticipate some of these liabilities, and I'd like to illustrate this in the next couple of slides. So the first of these is an anti-NGF antibody created by Mammalian from phage display. The original antibody was a pretty good antibody, had an affinity of 69 picomolar with good biophysical properties. The antibody, however, was affinity matured to improve the affinity. However, in so doing, various developability issues were introduced. Now, these issues are not obvious from simply looking at the melting temperature or even the yield that was produced in transient culture. Both the parent MEDI 578 and MEDI 1912 were, according to the publication shown here, expressed it in the territory of 200 microgram per mil in transient culture. However, when one started trying to work with the antibody, concentrate it up and so on, it became apparent that liabilities had been introduced. The new antibody suffered from self-interaction, resulting in precipitation at relatively low concentration and increased viscosity of preparations of this antibody. In addition, there was lots of non-specific interactions identified, either from just looking at cell binding or from immunohistochemistry, for example. And quite probably as a result of this low specificity, the antibody also suffered from a short half-life in vivo, whereas Medi578 had an 18-day half-life. Medi1912 had only 6 days half-life. However, the group were able to look closely at this antibody using hydrogen deuterium exchange, analysing aggregation propensity, and were able to identify 3 residues that they suspected were causing the problem. These were non-paratopic residues, so the expectation was the antibody could be fixed without affecting affinity, and that indeed is what they did by introducing the 3 amino acid changes shown here, and the new antibody was named Medi1912-STT, reflecting the amino acids that had been introduced. So we wanted to know if any of these problems, biophysical problems that this antibody experienced, would be reflected in terms of the presentation level when we put it into the mammalian display system. We also picked 2 other examples from the literature of antibodies where there had been a parental antibody with developability problems which had been fixed by mutagenesis, and one of these examples is an antibody from Centocor, an anti-IL-13 antibody described in this paper by Bethia et al., and also an anti-angiopoietin antibody, again from Medimmune, where there had been a parent with developability issues that had been fixed by mutagenesis. We took all 3 of these pairs and put them into the mammalian display system, and as you can see on this histogram shown here, that we could indeed see the differences in presentation level of the improved clone compared to the original parental antibody. So these differences that were being experienced in terms of self-interaction or other problems appear to be reflected by differing presentation levels on mammalian display, opening up, we believe, the idea of even incorporating this into the discovery process. So why would display on the cell surface help you to pick up some of these biophysical problems that antibodies may suffer from? Now, when we secrete an antibody from a mammalian cell, it goes out into the large volume of the tissue culture media. However, by mammalian display, we actually anchor the antibody on the cell surface and thereby can achieve relatively high concentrations in a short space of time. So to look at this, we've actually tried to quantitate the amount of antibody displayed on the cell surface, in this case using quantum simply cellular beads, which have a range of antibody levels on the surface and allow you to create a calibration curve as shown here, from which we can calculate copy number. And again, we can see from the 3 pairs that we've just discussed that you can indeed see a difference in a quantitative difference between the parental and the improved offspring. You can also see for a couple of well-behaved antibodies at the bottom here, ustekinumab and pembrolizumab, they were actually achieving surface display levels approaching 1 million copies per cell. So using these numbers and the fact that a hex cell has a diameter of 20 microns, we can actually calculate the volume that an antibody occupies on the cell surface, assuming that we have an antibody of 150 angstroms. Using these numbers, we've actually calculated the concentration approaching 13 milligrams per mil is achieved on the cell surface for an antibody expressed at 1 million copies per cell. So this is in fact above and beyond the concentration where antibodies with liabilities end up precipitating, and so we believe the system is working by exposing antibodies to a relatively high concentration, and any antibodies with issues will be manifest as low presentation levels. We use the MaxCyte electroporation system not only for making our libraries but also for transient expression either in HEC293 cells or CHO cells. And the table on the bottom right here shows the result of comparing the properties of the MEDI1912 parental antibody with the improved variant. And as I mentioned earlier, you can't really tell by looking at the melting temperature that one is improved over the other or that the parent was problematic. Similarly, the expression yields between the two aren't significantly different. However, again, in our hands, when we concentrate these up, we can see that the parental antibody does indeed precipitate, in our case at 1.4 megamil. As I've shown on an earlier slide, we can take these two antibodies and present them on the cell surface and can distinguish different presentation levels. So we then created, if you like, a model library by making a 50-50 mix of these two antibodies, introducing them into the mammalian display system. And after 12 days of drug selection, we've carried out flow sorting on the basis of FC expression, which is shown in the dot plot on the top left. So if we take this high expression subpopulation and analyse the sequences of that, we find in fact that the majority of clones within this population are indeed the improved MEDI1912 STT variant. In contrast, when we take the lower expressing population and sequence that, we find the majority in here are indeed the parental population. So this is giving us an indication that we may be able to use the mammalian display system in a real-life library setting to separate populations of cells on the basis of presentation level. Okay, so here we've gone to the same three positions that the MedImmune paper described, amino acids position 30, 31 and 56 on the VH gene, and we replaced the original hydrophobic codon with a randomization codon encoding 32 different codons encoding 20 amino acids. So in total there was something like 27,000 nucleic acid variants encoding something like 8,000 amino acid variants. This again was introduced into HEC293 cells by nuclease directed integration. After growing an antibiotic, the population was sorted on the basis of FC expression as shown in this flow cytometry dot plot. Again, the clones were recovered and were expressed and sequenced. And again, we were able to show that we had been able to create a number of improved variants compared to the parental clone. We can see, for example, some of the clones were able to precipitate up to 50 megamil without precipitation, while the parental clone was precipitating at 1.8 megamil. And again, using dynamic light scattering, we can see improved properties in terms of the size average and polydispersity of the clones. I'd now like to take another example where we've been able to take an antibody with poor biophysical properties and improve it using the mammalian display system. In this case, it's bococizumab, an anti-PSK9 antibody developed by Pfizer, and in fact this had gone all the way through phase 3 clinical trial before being pulled by Pfizer last year. This started as a mouse hybridoma, which was humanized, and then using phage display, the affinity of the clone was improved from 300 picomolar to 5 picomolar. Unfortunately, while focusing on improving the affinity, this antibody gained some liabilities from a point of view of development. And in fact, in this paper, there was this excellent paper from Adimab from a year ago, Jane et al. They looked at a whole panel of antibodies, something like 140 different antibodies that were at different stages of progress through the clinic, and they expressed them all and analysed their biophysical properties. And this particular antibody, bococizumab, scored badly in that assay. So in fact, there's a number of assays that were looking either for evidence of self-interaction, and you can see them listed here. I'm not going to go through them all. Other assays that were looking for low specificity of the antibody being tested, and again, bococizumab scored badly in a number of these assays. So we took this as an example, and in fact, it's quite possible that part of the problems that they encountered in the phase 3 clinical trial may have arisen because of these poor biophysical properties. It was reported that many of the patients ended up creating anti-drug antibodies. Something like 50% of the patients within a year had an immune response. So we took bococizumab and presented it by mammalian display, shown in the next slide. We can see here, by comparing bococizumab to the original humanized version, that the display levels was significantly less than found on the humanized version, again suggesting that this antibody had potential, but problems were introduced through the affinity maturation. So we again looked at bococizumab and identified a number of residues that we felt may have contributed to the problems. So we identified 4 residues in the VH region and 3 residues in the VL, and we constructed a library where we mutated in these positions, and we selected then for improved FC presentation on the cell surface. So this next slide shows us the result of that. So let me explain that when we do mammalian display, there's an initial transient expression for a few days. Then after about a week, that transient phase passes. We still see a negative population of cells, as I've mentioned. 5% of the 1 to 5% of the cells actually take up and express the antibody stably. So we have to go through a process of drug selection, and that's why you can see a negative population in the top 4 examples, the top 4 histograms here. But you can nonetheless see that bococizumab is giving a lower level of expression than its the humanized parent 5A10, and below the 5A10 you can see the profile from the library, and already one can see that there is the appearance of higher expressing clones, in this case, or higher presenting clones. The library represented on line 4 was then passed through max speeds and selected either on the basis of FC expression or on the basis of antigen binding. So these cells were then cultured for an additional 9 days. So if we then take these populations of max sorted cells sorted on day 7, grow them for a further 9 days and carry out flow cytometry, flow sorting, we can again see the population is still largely double positive, as one might expect. If we then sort here on the basis of FC expressing using flow sorting, we can then recover the antibody genes, sequence them, identify unique clones and express those. And when we do this, when we express them and then characterize them by AC SINs, which is a method that's useful for looking for self-interaction, we can see already that 84 out of 89 clones are better than bococizumab, and indeed the additional 5 clones here, when we purify them and express them at normalized concentration compared to bococizumab, we find that they too are improved over the parental clone. So we then took a number of the clones selected on the basis of high-level presentation on the cell surface from the library, and expressed them and purified them at a higher level, and ran them again through AC SINs, confirming that there was indeed reduced self-interaction using the purified material compared to the parental bococizumab antibody. And in addition, we've ran them on HP size exclusion chromatography, and we see an example of some of the clones here. So in the dotted line you can see alirocumab, which is a competitor anti-PSK9 developed by Regeneron and Sanofi, and that has very nice properties in terms of its profile on size exclusion chromatography. In contrast with bococizumab, you can see that there is interaction with the column, causing this retention and a slower release from the column, and a broad peak. In comparison, the 5A10 humanized antibody from which bocakizumab was derived gives a better looking profile than bocakizumab, and here we can see one of the clones that we've selected by mammalian display, again showing these improved properties. So in summary, we believe mammalian display is a very powerful technique for screening millions of antibody clones for their binding properties, with the advantage of being able to work in an IgG format and also to work within the cell type that may eventually be used for production of the antibody. This deep screening that's enabled then allows us to potentially carry out affinity maturation much more efficiently, or simply to take outputs from other techniques such as phage display and screen them in depth. The approach we've taken uses nuclease-directed integration to put a single antibody gene into a single locus within the genome, and in so doing we've basically achieved transcriptional normalization, and therefore any differences that we see in terms of expression are down to the properties of the antibody itself. But apart from just thinking about expression, we believe that the system is actually letting us see a little bit more into the biophysical properties of the antibody. In particular, I think it's important that we can achieve high concentrations of the antibody on the cell surface, and therefore in this very early discovery step we're actually achieving the sorts of concentrations that are often required to generate high concentration of antibody, for example for subcutaneous administration. Thus we believe the system has the potential to pick up and some of these issues much earlier on, and potentially avoid clones like this with biophysical problems being selected for further development. And in addition, there is also the opportunity to take antibodies that have problems, create libraries from them, and basically select improved variants. So in other words, to fix broken antibodies, as it were, as we've shown in the examples here already. And at the end of the day, that really translates into large savings in time and money if one can choose the optimal clone at the point of discovery, rather than having to screen lots of clones later in the discovery process. I'd like to finish by thanking the people involved in this work over the last 4 or 5 years. Mike and Kotai have been mainstays of this project throughout, ably assisted currently by Ed, Maheen, Rachel, and prior to that by Yanchao, Sophie, Raj, and Johanna. And these people listed at the bottom have also made important contributions to this work. So it simply remains for me now to hand back to the moderator to introduce the next speaker, and thank you for your attention.
Thanks, John. That was a really excellent presentation describing the power and advantages of a mammalian display for streamlining the therapeutic antibody discovery process. Before we move on to the next presentation, I want to remind the audience once again to submit questions for our Q&A session at the end. We'll try to answer as many questions as we can, so simply type your question into the box below the presentation screen and hit submit. All right, now that that's all squared away, Payal, take it away.
Hello, I'm Payal Roy Chaudhary, Field Application Scientist at MaxCyte. I have been providing technical support to Dr. McCafferty's group at IANTAS. I'd like to spend a few minutes to tell you about MaxCyte and the unique approach that we have taken to cellular transfection. Our technology was developed for clinical use, and therefore our focus has always been on providing a safe, non-viral approach to loading molecules into challenging cell types with high efficiency while maintaining high cell viability. MaxCyte's cell loading technology is based on electroporation. We suspend the cells in physiologically balanced conductive buffer with relevant loading agents and then apply a series of electric pulses that create a temporary perturbation to the cell membrane, allowing molecules of almost any size to enter the cell very rapidly. Our motto is any molecule, any cell, any scale. We can efficiently load DNA, mRNA, siRNA, and even proteins into many different cell types. The technology can be used with a wide range of primary cells and cell lines, including insect cells. One of our technology's most unique features is scalability. We can transfect small number of cells by static electroporation, which takes only a few seconds. Large number of cells between half a billion and 200 billion cells can be transfected by flow electroporation. The MaxCyte's delivery platform has been adopted by many pharmaceutical companies, biotechnology companies, academic institutions, and other organizations to develop a range of therapeutic modalities. We can generate modified cells for autologous and allogenic cell therapies. We enable large-scale manufacturing of viral vectors and VLPs for vaccine and gene therapy applications. We can produce high-titer antibodies and other protein-based biologics in cell types that are relevant for bioproduction. We can also express complex drug targets such as ion channels and GPCRs in large number of cells for cell-based drug screening. We have abundant experience in working with gene editing tools, especially working with CRISPR-Cas9, TALEN's zinc finger nucleases, and meganucleases. We have a number of collaborations with Harry Malak's group at NIH, from which we have publications available in public domain. MaxCyte offers two different instrumentations for basic research, bioproduction, and high-throughput screening applications. The MaxCyte XDX instrument can transfect as few as 500,000 cells or up to 20 billion cells at one time. Smaller cell numbers up to 700 million cells are transfected in a few seconds. Large number of cells flow through the electroporation chamber. During electroporation, cells are suspended in MaxCyte's chemically defined electroporation buffer at high density, typically 100 to 200 million cells per ml. Thus, 20 billion cells can be suspended in 100 ml of electroporation buffer and processed through the XDX instrument in less than 30 minutes. Our VLX instrument can process 1 liter of concentrated cells or up to 200 billion cells in a single transfection run. I also want to point out that all transfections take place in sterile single-use chambers called processing assemblies. We have different sizes of processing assemblies to accommodate different number of cells. The MaxCyte technology is very simple to use. Users do not make any adjustment to the electrical parameters. They simply select a protocol optimized for their cell type from a drop-down menu. Currently, we have optimized protocols for over 80 cell types. If your cell of interest is not on this list, we also provide 10 optimization protocols ranked from low to high energy. That should allow you to easily find conditions that provide good transfection efficiency and viability with almost any cell type. In conclusion, I'd like to highlight the key benefits of MaxCyte's scalable cell delivery platform. With our system, you can work with the appropriate cell types. User can produce a wide range of molecules. High productivity stems from high transfection efficiency and viability. MaxCyte's electroporation platform is much more consistent and scalable than chemical-based transfection methods. Higher productivity in turn allows customers to drastically shorten development timelines. And with this, I'll end my presentation and hand over to Jen for Q&A.
Great presentation. Thanks very much, Payal. I think our audience can now fully appreciate MaxCyte's unique technology and approach towards transfection, which should save investigators plenty of time, resources, and money. So before we start the Q&A session, I want to remind everyone that this is their final chance to submit questions for our speakers. So hurry up and send them in now. Just simply type your question into the box below the presentation screen and hit submit. Okay, it looks like we have a bunch of really great questions that have already rolled in. So let's get to the Q&A session. We'll try to get to as many questions as we can, so if you still have a question, send it on in and we'll try to get to it. Give us a few moments on our side to get everything situated, and we'll transition into the Q&A session presently.
All right, so let's start the Q&A. John, first question is going to be for you. Basically, I saw that there's a press release this week that you've done a deal with Sanofi, and I assume that the technology is ready for prime time. So my question is it, and how long does the project take compared to, say, antibody discovery by phage display?
Okay, thanks very much, and thanks everyone for joining the webinar today. So yes, we did indeed have a press release out this week. So to answer the question, is it ready for prime time? Absolutely. We've had a team working on this technology for about 5 years now to ensure that we hit the ground running. And so we've invested quite a bit of time and effort over a period of time just to make sure that when we did go out, we were match fit, as it were. Again, one of the drivers for us was to make it actually timely to have it such that we could generate antibodies in the same or better timescale than phage display, and we have indeed achieved that in terms of generating a panel of IgG formatted antibodies. I guess with phage display, powerful technique as it is, at the end of a discovery phase, we still have to reformat into IgG format, but of course that's the standard format we're using for mammalian display. And on top of that, at the end of the mammalian display process, we will have screened millions of candidate antibodies rather than hundreds, so the depth of that screen is increased by 3 or 4 orders of magnitude. So the other thing I would say is it's actually particularly powerful for affinity maturation programs, since we can screen potentially all the variants within a library and at the same time distinguish between expression level and binding level and thereby infer something about affinity. So again, as far as affinity maturation goes, we think it's quicker and more powerful there as well. Thank you.
All right, thanks, John. And just one more follow-up question to that. How does the flip-in system compare with your nuclease-directed approach?
Okay, well, actually, both the nuclease-directed and the flip-in approach are both looking to generate single transgenes per cell in a single fixed locus. The flip-in system, which is sold by LifeTech, is fundamentally built for creating relatively rare events, which is fine if you're trying to make a single cell with a modified gene, for example. But it's based on, it uses a crippled flip recombinase system. And in our hands, we find actually in direct comparisons, if we use the wild type flip-in, it actually kills cells. And again, the flip-in system recommends using small amounts of donor DNA. And if you increase the amount of DNA, you certainly increase the number of colonies, but you also increase the background from random integration and potentially multiple integrations. And so in side-by-side experiments, we found lower specific transformation efficiency from the flip-in system and a lower fraction of the cells that are actually expressing antibody following drug selection when we directly compare the flip-in system with tail nucleases and meganucleases in the same cell line. Thank you.
All right, thanks, John. Payal, we have a question for you. One of our audience members would like to know how easy is the MaxCyte platform to set up and use for antibody development projects?
Thank you. Thank you for the question, and thank you for listening to the webinar. So yes, the MaxCyte system requires no specialized media or culture conditions. What we do recommend is a single chemically defined buffer, and it is compatible with a wide range of cell types, including multiple lines of CHO cells. Also, optimized electroporation parameters are already programmed into the software, so the user does not need to optimize any of the electroporation parameters for the actual transfection process. And MaxCyte's XTS is a benchtop instrument that can be set up within minutes, and it is easily integrated to any antibody discovery and development programs. Thank you.
Great, thanks, Payal. John, one question for you. Is the mammalian display technology only available through Iontess, and how accessible is it to other researchers?
Okay, so the problem to solve is how to make big libraries in mammalian cells with a single antibody gene per cell. And a number of companies and labs have taken different approaches to solving that problem. We have taken the approach of targeting all of the genes into a single locus in the cell. And we think that this transcriptional normalization that we achieve makes for cleaner results, and particularly when it comes to looking at the level of expression and or the developability comparisons. So as far as the technology goes, we are making it available now through collaborations or contracts with other companies, where we will either run programs here in-house for the partner, or in some cases will be affected by technology transfer. I mean, in terms of the timing of running a certain in-house program, for a reasonably straightforward antigen, we're usually talking about 4 months to generate a large panel of IgG formatted antibodies with particular specificity and affinity requirements. And again, because we're working in a flow cytometry system, apart from being able to screen tens of millions of clones in a relatively short time, there is, of course, the opportunity for multi-parametric sorting, so we can screen out cross-reactivities we want or that we don't want, as well as building in these aspects of developability that we spoke about. Thank you.
Great, thanks, John. The next question actually is probably one that both Payal and John can weigh in on. Our audience member would like to know what made using the MaxCyte platform particularly unique for mammalian display versus other commonly used methods for transfecting eukaryotic cells.
Okay, shall I pick that one up first, Payal?
It's left to you, John.
Yeah. So yeah, and again, for us, the challenge here is how to make large libraries, as large as we possibly can. And we looked at a number of different systems. The thing that drew us to the MaxCyte system was, first of all, the high transfection efficiency that's achieved, but also the fact that it's a system that was built towards increasing cell survival. As I understand it, this was originally developed for clinical applications where you really needed, 95+% of your cells to be viable at the end of the electroporation, and that's not the case with most other systems. So that, for us, was the big driver. And of course, the other benefit for us with the system was that, well, actually, I've been quoting you can transfect 2 times 10 to the 10 cells in half an hour, but actually I see from Payal's slide I'm an order of magnitude out in the wrong direction. So for us, viability, the ability to transform huge numbers of cells and therefore build massive libraries. At the moment, we are working with libraries maybe around the sort of 10 million mark, although again with the MaxCyte system, there is a potential for us to have libraries up in the multi-10 to the 8s if we so wish. Payal?
Yeah, so I'll add to John's comments there. So the value proposition of our technology is one could effectively transfect cells with very large molecules or also with multiple loading agents and achieve high transfection efficiency as well as viability. And one of the unique features, as John highlighted, is the scalability of our platform. So one could transfect from 0.5 million cells up to 20 billion cells within less than 30 minutes. So, yeah.
And actually, I would add that we initially bought it principally because of our interest in making large mammalian display libraries, but it's now also a workhorse for just general antibody production within the lab. And again, over the years, we've focused mainly on HEC293 cells, which are very easy to transfect. But again, with the MaxCyte system, we're moving more now to working with CHO cells, even for basic production of IgGs. Thank you.
All right, thank you very much, guys. John, another question for you. One of our audience members would like to know, how are the antibodies tethered to the cell surface, and can this be switched to produce secreted IgG?
Yeah, so the original system as we built it uses a PDGFR transmembrane domain, so the antibodies are tethered in that way. We built the system such that transmembrane domain was represented within an exon that we could, if necessary, remove. And downstream of that exon, there is a follow-on exon that encodes a termination of that antibody sequence. What we found in practice was that actually we were achieving some soluble expression even without removing the exon encoding the transmembrane domain, and we believe that's down to alternate splicing. In the follow-on system, we've played around with the way that we tether it, and we now have a system where potentially we can have a greater amount of secreted antibody produced and some level of control in the extent to which it's presented or to which it's secreted. So, and actually, I think that same person asked another question. Yeah. So. Sorry, I don't mean to take over the moderator's job here, but I see that it's the same person asked the question by percentage stable integration. Do you mean post-selection, i.e. 95 to 99% of the cells that grow through selection do not express antibody? No, absolutely not that. What? In fact, I think on one of the earlier slides we showed that. So no, when I'm talking about 95 to 99% that don't, I mean that's basically 1 to 5% of the cells will actually integrate the antibody and give rise to a drug-resistant clone. So by the time we've done drug selection over 2 or 3 weeks, we find that the vast majority of cells are now all expressing an antibody. I think, have I answered that? Yeah, that's it.
All right, thank you, John, and stay with me because we have time for one last question. Audience member asks, can the enriched antibody population be from any source, such as phage, yeast, mouse, humanized mouse, etc.?
Yeah, so here at IONTAS, and in fact in the days before that, both at the Cambridge Antibody Technology, the Sanger Institute in Biochemistry, we've always used phage display, and we're great fans of the technique and technology. But of course, the input antibodies can come from any source, from any direction. And as I said earlier in the presentation, sometimes people go on about antibodies from phage display being difficult to work with, whereas in fact what phage display does, it gives you enormous power in terms of making large libraries and in terms of driving selection towards high affinity. But with that power comes the potential to introduce liabilities, and we saw 2 examples of that actually in the presentation, the anti-NGF antibody from Mammalian and the anti-PSK9 antibody from Pfizer. And again, I would make the point that sequence liabilities or developability liabilities can come from any source, and that includes so-called natural sources. And in fact, our experience of making mouse antibodies by going to patents and recovering antibody sequences is that actually most of the time they express less well than we're used to seeing with the human antibodies from the phage system. So yes, in short, antibodies can come from any source.
All right, thanks, John. And actually, we have time for one more question for Payal. Payal, what's the advantage of using electroporation compared to a transfection reagent?
Thank you. So, to talk about our delivery platform, so we were specifically designed for clinical applications. So we are a spin-out of Center of Blood Research from Harvard University. So in principle, we answered the difficult question first. So we met the stringent criteria of stem cells having high cell viability as well as high efficiency. So when we compare ourselves with other transfection methods which are available, we offer much more delicate conditions, if I say it that way, to get much more high transfection efficiency as well as viability, where we are able to load the molecules, whether they are large or whether there are 2-3 plasmids, we can do it very effectively. Thank you.
Thank you very much, Payal. And with that, we've come to the end of our webinar. I'd like to remind everyone that the webinar will be archived on our site at www.genengnews.com. So if you missed any parts of it or want to watch it again, you can do that, or feel free to forward the link to your friends and colleagues, which we always recommend. I'd like to thank John and Payal for their informative presentations, and I'd like to thank the audience for their attention and very astute questions. And a very special thanks to MaxCite for sponsoring this webinar. So hopefully we'll see you again at another Gen webinar very soon. Goodbye for now.